So today we're going to hear from Anastasia Yendiki. And so Anastasia got a degree in electrical engineering in Greece, where she's originally from. And then she went on to get a PhD in the University of Michigan. And then she went to do a postdoc at Massachusetts General Hospital, which is actually where I met her. And now she's an associate professor in Mass General Hospital with appointments in radiology at Harvard as well. So Anastasia's work looks at white matter connectivity in the brain and specifically tries to build computational models to tease apart white matter bundles. And she uses high resolution data to help inform low resolution data such that it can be useful for everyone and for anyone's scans. So she is the author and founder of TRACULA, which I'm sure she's going to talk in some depth about. Yeah. And it's a free and available software suite. So if you're interested in this type of stuff, you should definitely download it. She's also really good on the message board, so if you have questions.
I try. I don't know if I'm very good, but I try.
And apart from all this amazing, innovative, and translational stuff, she's also dedicated to open science. So like I mentioned, all of her software is online and available. But she's also an amazing flamenco dancer. So. And she was also recently, I just found out, was featured in InStyle's Badass Women because she is in fact very badass.
Thank you.
So without further ado, let me yield the floor to her. So she's going to talk about mapping the wiring of the brain with diffusion MRI, current tools, and future prospects.
Thank you very much for the kind introduction, kind and slightly embarrassing. And thank you very much for inviting me. This is my first visit to OSU, even though I've heard quite a bit about you guys. As you heard, I was at grad school at U of M. I have to say you look less evil than I had expected, so that's a surprise. But thanks for having me, and feel free to interrupt and ask questions at any point. So why do we use diffusion MRI data? It allows us to map the white matter pathways that connect different brain regions. So the basic idea is that we collect a series of diffusion weighted images. Each one of these images tells us something about how much diffusion of water molecules there is in a particular direction, and in another direction, and in another direction, and so on. And from that, we can infer a bunch of useful measures. Markers like diffusivity or anisotropy maps can be used as indicators of myelination or coherence of white matter bundles. And in addition to that, we can extract these vector maps that show the preferential direction which is easier for water molecules to diffuse. And tractography is the methodology of following these vectors around the brain, connecting the dots to reconstruct white matter tracts. And once we have these tracts, we can then localize the different changes that we see in the anisotropy, diffusivity, etc., maps in terms of specific brain pathways that they affect. So there are two ways of going from running tractography to connect those dots in the brain to extracting anatomically meaningful bundles that we can use to test hypotheses. One is atlas-based. So when you have a specific bundle that you're interested in that we know exists in everyone's brain, like the uncinate fasciculus, the cingulum bundle, etc., you use some kind of prior anatomical information to reconstruct these predefined bundles automatically in a set of subjects. The other approach is data-driven, where you don't use any a priori information about what bundles are actually there, but you run whole brain tractography seeding everywhere in the brain, and then you use a clustering algorithm to group these tracts based on their similarity. This allows you to perform more exploratory studies and explore the structure in the data rather than imposing some pre-existing knowledge about white matter organization. And I will start with discussing our tools in the first category. So when tractography was first introduced in the late 90s, early 00s, the simple way in which people came up for reconstructing known white matter highways, like the big bundles like the onset fasciculus, the corticospinal tract, etc., was to basically define each of these pathways based on 2 to 3 regions of interest that pathway is known to go through in the brain. So you run tractography, you draw these ROIs that you believe define the specific bundle that you're trying to find, like in this case, the cingulum bundle, and then you hope that there are some tracts that are found to be going between those 2 ROIs that you drew. So there are 2 issues with that. First of all, drawing these things in every single subject takes time. So it's not automated, or it wasn't at that time. And then it's not robust, because you're not guaranteed, even after you do all this drawing work, that you will get tracts that go between the 2. So then people came up with methods to automate this approach, which basically consisted of doing the same thing, running tractography, but now using ROIs that were not manually drawn, but they were mapped from a template to your subject. And then again, hoping that there are some tracts that go between both regions of interest. And most of the automated tractography tools that are included in many of the toolboxes out there belong in this category. So this solves the manual labeling problem because it's automated, but it still does not solve the robustness issue. So you're still not guaranteed that you will find in your individual subject the tracts that exactly represent the bundle that you're looking for. So it's helpful to go back and look at how the people who were originally working on defining these bundles actually defined them. And this is anatomists who did this work not based on diffusion MRI, but based on chemical tracing in animals. So if you open any anatomy book, you will not find that axon bundles are defined based on some MNI coordinates or regions of interest. You will find that they are described in terms of the structures that they go through or next to, and their relative position with respect to these structures. So for one random axon bundle here from the Fiber Pathways of the Brain book, you'll see that this bundle goes below and medial to the claustrum. It goes caudally until the most caudal level of the LGN, medially to enter the thalamus, and so on and so forth. So you'll see that there are absolutely no template space coordinates or regions of interest that are defined here. Instead, the bundles are defined based on their relative position with respect to other anatomical structures. Now you might say, well, this difference in approach is because anatomists are old school, and so they should just get with a program and start using templates like we do. But I will counter that maybe there is something to this approach that we can learn from. So maybe this is more robust and it's easier to generalize between different brains within species, but even across species. So you could take these rules and apply them to find the same bundle in a human, even though this was defined on a macaque brain. Furthermore, what we learned by going back to the source material, these anatomical tracer studies, and in this particular case work from Suzanne Haber's lab at the University of Rochester, is that these large highways, like the onset fasciculus, the cingulum bundle, etc., they're not monolithic structures. They're not all a bunch of wires that go from point A to point B. Instead, they serve like highways, and smaller axon bundles will merge on and off those highways at different positions along the trajectory of this highway. Furthermore, her lab has shown that in some of these large bundles, like the internal capsule, the corpus callosum, etc., there is very specific topography of those smaller axon bundles, depending on where those bundles originated in the cortex. Not all axon bundles are organized that way topographically. For example, the onset fasciculus seems to be messier, but many of them do. And so the question then becomes, can we reconstruct these kinds of topographies in the human brain using diffusion MRI? And first of all, why would we want to? Well, this can be clinically relevant. So this is data from Amelia Versace and Mary Phillips at the University of Pittsburgh, who also collaborate on this project with Suzanne, showing differences in fractional anisotropy, one of those diffusion-based biomarkers that I mentioned in the beginning, in bipolar disorder patients versus controls in the internal capsule. And so they found that the differences were detectable when you only looked at the part of the internal capsule that, based on the rules that Suzanne's lab developed on monkey brains, are connected to the VLPFC. And if you average the FA everywhere in the internal capsule, you would lose the effect. And this makes sense in terms of functional differences that have been found in these patients in the ventrolateral PFC. So being able to reconstruct these topographies of the smaller axon bundles within the larger bundles, based on where these axon bundles originated in the cortex, has implication for specificity and interpretation of imaging findings, as well as helping with selection of therapeutic targets. So then the question becomes, well, how reliably can we find these topographies in human brains? And so Suzanne put some tractography seeds in the areas where she had injected the macaque brains to find these organizational rules. And we were able to do this using the arguably the highest quality diffusion data that are publicly available at the moment, which is the MGH HCP B equals 10,000 data. So this is just 35 subjects collected with very high B values and very high resolution. And so she was able to reproduce some of these topographies. So this is an example of tracks that originate in 3 different cortical areas, going from more dorsal to ventral, and then showing that when these 3 bundles reach the internal capsule, they maintain this topography. So how easy is it to see this in individual human subjects using that, very high quality data? So this shows pairs of 2 bundles in each of the plots. One is the green one is the red one. And the names there are just the Brodmann areas where they were seeded. And the crosses are where inside the internal capsule these tracts ended up based on what cortical area they were seated in. This shows those centroids of those bundles in MNI space, where subjects were registered after the tractography was run in their native space. They were registered to the MNI template with nonlinear registration. What you see is that across individuals, many times the green from one subject and the red from the other subject will overlap. And some of those are easier to discern, and some of them really have a lot of overlap. So it would be very hard to predict, even with nonlinear registration, the location of these bundles in individual subjects based on template-based coordinates. But now let's take the pair of 2 crosses, the red and the green from each subject, and move them so that the red is always in the origin. Then the green consistently will fall in the same quadrant, which means that the relative location of these bundles is preserved. So which one is medial, which one is lateral. And this suggests that it is much more robust to try to reconstruct the organization of the white matter based on the relative positions of structures to each other, right, rather than based on absolute coordinates in a template space. And luckily, over the last few years, our group has been developing methods to do exactly that for tractography. And TRACULA was the effort to formalize this in order to reconstruct white matter pathways automatically in routinely acquired clinical quality diffusion data. So as opposed to the multi-ROI method that I described earlier, instead of just running the tractography and then drawing the ROIs or taking them from an atlas completely independently, the information about the relative position of these bundles with respect to their surrounding anatomy is included in the tractography step itself. So rather than just having rules based on deterministic ROIs, we use a bunch of subjects, a bunch of cases where these tracts were labeled manually, and we learn by walking along each of these tracts, who is my neighbor, medially, laterally, rostral, caudal, etc. And then we build a prior probability of the anatomical neighborhood of this tract. In this case, it's the anterior thalamic radiation, as a function of where along the tract you are. So not just the endpoints, but everywhere along the tract. And so we fit the tracts then simultaneously to the orientations from the subject's diffusion data, as well as this prior information on what parts of the anatomy this particular tract is likely or less likely to go through or next to in each direction. And the good thing about this is that because we're using relative locations in individual space, rather than enforcing some MNI coordinates that all of our tracts of all of the subjects have to go through, we are quite robust to whether we use healthy subjects to label these tracts in, or whether we use patients as well. So we showed in the first TRACULA paper that the accuracy of the automatic reconstruction when compared to manual labels of the same tracts did not depend on whether we included patients or just controls in the data that we use to train the algorithm. And that's because we are only assuming that the tracts will go through the same general parts of anatomy. We're not assuming that the tracts have the same size or the same shape or the same integrity in the patients that we're analyzing as in the controls that were used to learn the shape of those pathways. Then another benefit of this approach is that it's easy to generalize to other designs like longitudinal data, for example. A problem when reconstructing white matter tracts in longitudinal data with conventional streamlined tractography, where we just run the tractography on the data from each time point independently, is that as, let's say, we have degeneration, we might get less and less of the tract at each successive time point. And then so it's not like this part of the tract doesn't exist. It's not like somebody went in and cut the tract with scissors. It's just that the anisotropy gets lower here, and so the tractography doesn't know how to proceed, and so it just stops. So in this case, we would be comparing apples and oranges between the different time points. You can imagine that if it's a developmental study, this could go the other way around. So you could have only parts of the tract in the younger subjects and more of the tract as there's more myelination and it's easier for the diffusion tractography to find more of the tract. So you do not have point-by-point correspondence to compare the tracts point by point across time points. And you may underestimate changes in anisotropy or diffusivity exactly because you're missing the part of the tract where most of the changes occur. So with the TRACULA framework, we can easily generalize it so that it uses, because it's a Bayesian framework, we can generalize it so that it uses the data from all the time points all at once. So we use the diffusion series from each of the time points of this particular one subject, plus the structural segmentations from all of the time points of the subject. And we we reconstruct the tracts jointly using all of these data. And this ensures that there is exact point-by-point correspondence across all the time points. And it is unbiased in the sense that it treats all the time points the same way. One way in which you could bias the analysis is if you did the tractography in the baseline scan only, and then you registered all the follow-ups onto the baseline scan. Because you are analyzing the follow-up and the baseline in a different way, you may introduce differences because of the different processing that are not actually there. Unlike that approach, this is unbiased because all the time points are treated the same way, and you can have any number of time points. And we showed that this, when you compare it to the cross-sectional analysis, where you analyze each time point separately as if it were a cross-sectional time point, doing the joint reconstruction using the Bayesian framework in TRACULA, using all the time points at once, improves both test-retest reliability in healthy subjects, as well as power to detect subtle along the track changes, in this case in a data set from Huntington's disease patients. Some things that we're working on right now. Somebody was asking about the HCP data. So we are working on redoing, and by we I should say my postdoc, Chiara Maffei, who's doing all this work with a little army of undergrads. So redoing the manually labeled tracts that we use to train TRACULA based on the extra high quality B equals 10,000, 1.5 isotropic resolution HCP data. So now we are able to get much nicer models. This is an example of the 3 branches of the superior longitudinal fascicula, so 1, 2, and 3. When you compare it to the much lacking reconstruction manual labeling of the same tract in more conventional data. So the idea here is to train TRACULA with this high quality data and improve the reconstruction that you can get in conventional data where you don't have your own connectome scanner and you're simply collecting the data with a lower B value and lower spatial resolution. Yes?
How are how do you know whether the tracks are labeled correctly? Like, what are you basing it on?
So we are, and by we, again, I mean Chiara, she's going back to the anatomy and looking at, anatomical studies about where the terminations of these tracts are. And in some cases, there are still controversies about some of these things. So we have to go with basically whatever is generally accepted. And of course, different anatomists may have slightly different definitions about what the SLF1 versus 2 is, but we have to kind of go with a consensus. Yeah. So it's basically, instead of just using those like the 2 ROI methods, because those methods were developed for much lower quality data that we had like 10 or 15 years ago, we actually now have to go back because we can get much more detail now. And so it's not enough to just, draw the 2 regions of interest to label these tracks manually. So we have to revise the protocol in many cases. And, this has been a process that has been taking like a couple of years.
I have a question.
Yes.
So I was curious, in the study that you did with individuals with schizophrenia and controls, I'm curious in these methodologies, how do you account for lesion load? So when there are white matter hyperintensities or lesions present in the brain, how does that impact the reconstruction of these tracts?
Yeah, so that's a great question. So actually, if you, I have to go way back. Okay, so if you were doing if you were using one of those multi-ROI methods, and if there were lesions somewhere in between here, it could be that you might not find anything that goes between the ROIs because the tract stopped. But with our approach, because we're not using that sort of conventional streamline tractography, but we are modeling the entire tract as a curve and trying to optimize the shape of that curve. We actually look at how well that curve fits the data everywhere along the tract, both the diffusion orientations and the information about the surrounding anatomy. So even if there is a lesion somewhere in between there, it's not going to affect the overall fit that much. And actually there was a visiting fellow in the lab over the last few months who applied TRACULA to subjects that had vascular disease type lesions. And so it's successful with that kind of subject exactly because the approach is different to just kind of like walking around and hoping that you get all the way to the other end. And we are also making sure that the new atlas contains many more tracts and covers much more of the white matter. And I should say that the new features that I mentioned will hopefully, fingers crossed, be included in the upcoming release of FreeSurfer that is planned for February-ish. I'm not sure if I'm supposed to say that, but Yeah, you didn't hear it from me. So using this kind of approach where we label the tracts in really high quality data and then apply the reconstruction to lower quality data, we compared how the TRACULA approach does in this task to the conventional multi-ROI approach, where we get our regions of interest by labeling the tracts in high quality data, and then we just map those from a template onto the individual that has the lower quality data and compare that to training TRACULA on the manually labeled tracts from the high quality data and then applying it to low quality data. So this shows an example of those three bundles that I showed you earlier, the three branches of the SLF. This is a manual labeling in the high quality B equals 10,000 data of a single individual from that data set. This is running TRACULA in using only the B equals 1000 shell from that same individual. And this is using the multi-ROI approach on the B equals 1000 shell. So you see that with this approach, because we have used the training data from the other high quality data sets, we get much closer, even with only the first shell, to the original manually labeled tracts in the much higher B-value data. This quantifies what I just mentioned. So it's the accuracy as in distance between manually labeled tracts in high quality data in B equals 10,000 data and automatically reconstructed tracts in B equals 1,000 data. So big distance is bad and small distance is good. So with the TRACULA approach, we get much better accuracy. We have also extended this to infants. So there is an infant stream coming up next, infant stream for free surfer reconal as well as for TRACULA. In this case, it's called Traculina, tracts constrained by underlying infant anatomy. In this case, for that first paper that we did, we trained on data from both full-term and premature neonates. And again, a similar approach to what I showed earlier for the adult stream. We showed that we could reconstruct the tracts in each population independently of whether that population had been included in the training sample or not. And we also showed, thanks to various hoops that our reviewers made us go through, that the approach is pretty robust to having errors in the segmentation. So we actually perturbed the segmentation labels and ran the algorithm again, or in registration errors. So because all that we use from our training data is the relative positions, is someone to my left, to my right, etc. It doesn't require very high accuracy for the segmentation or good inter-subject alignment at all. Now, all the stuff that I mentioned so far was the atlas-based approach, where we are interested in reconstructing specific known pathways like the uncinate, the cingulum, etc. And then I mentioned that there's also the data-driven approach where we do not have a hypothesis about a specific bundle, but we run whole brain tractography and then cluster the whole brain tracts into groups based on their similarity. This allows you to explore more, to perform more exploratory studies in the whole brain. So it turns out that this approach of using, of defining bundles based on their neighbors rather than based on MNI coordinates can be used for this approach as well. And this is a more recently developed tool that is also going to be in the next release. So the conventional way for grouping tracts to each other in whole brain tractography data is based on their distance. So if two streamlines are close to each other, they're considered to be similar and more likely to be grouped together. We have used an anatomical similarity, which throws away the spatial coordinates of the streamlines and instead compares whether they have the same neighbors. So we build a histogram for each of the streamlines from the whole brain that tells us how often they go to the left, to the right, etc., of each of the free surfer segmentation labels, and we cluster streamlines together if they go through the same parts of the anatomy rather than just based on whether they're close to each other. And you can imagine that there are situations where two tracks are close to each other, but they end up in different cortical terminations, etc. Like, for example, those sub-bundles that are inside the bigger highways, where simply grouping them by spatial distance would not be informative. And we have shown that using this anatomical similarity rather than the conventional distance-based similarity leads to clusters that are more similar to manually labeled bundles, even though we have not included any training data from manually labeled bundles here. We only use the subject's own segmentation to cluster the subject's own tracts. But because we use a similar philosophy as what anatomists would use to define that two axons are in the same bundle, based on whether they go through the same parts of anatomy, we end up with things that look much more like anatomically defined bundles. And the clusters are also more similar across individuals, as we've shown in a large lifespan data set going in ages from 9 to 90, even though we do not use any inter-individual registration, or actually because of it. So if you had to match clusters, track clusters between subjects with the distance-based approach, you would have to map all of them into a template space and hope that this point-by-point correspondence across the subjects in template space is accurate. Here, because we're matching them based on these histograms of their neighbors, we can compare these from different subjects without co-registering them. And so it turns out that doing our clustering this way and matching across individuals based on the anatomical neighbors of the clusters leads to better fitting of lifespan trajectories of diffusion measures. That work is in review at the moment. Now I'm going to shift gears a little bit. And so let's say we have all those tools. We get measures from all these different bundles that we've defined and the different approaches that I mentioned earlier. How do we use this for predicting individual clinical trajectories, and how do we combine it with functional data, which is something that I'm sure some people here may be interested in. And I have to apologize, the next two slides, I made them like, I don't know, 10 minutes before the talk, based on condensing 12 slides that Sue sent me half an hour ago into one. So I hope this will make sense. So this is data from the BANDA study that we have been conducting over the last 4 years. That's the Boston Adolescent Neuroimaging of Depression and Anxiety. It's one of the projects from the second phase of the Human Connectome project, one of the connectomes related to human disease projects, which means that it will also become available through the Connectome database. And it's a collaboration with many different institutions and many different investigators, and led by Sue Whitfield-Gabrielli and John Gabrielli. And this is looking at adolescents between ages of 13 and 16 at baseline. Apologies again for the small, size of the figures, but I'll lead you through it. So these are, the data collection focused on patients rather than controls to be able to capture more of the variability in terms of depression and anxiety symptoms and their comorbidity. So when we look at the different clinical scales, there is quite a bit of spread, both inside with the control group, but especially across the patient group. And also, when looking at change of their symptoms within 1 year from the baseline visit, for the controls, which is the green, they stayed pretty constant, but there's quite a bit of change in terms of the trajectories of these symptoms for the patient population. And so perhaps unsurprising, if you simply did a group comparison of MRI measures, that would not be very informative. So if you can see it here, the green is fractional anisotropy of the uncinate fasciculus from the controls, and the blue from the patients. So you can see that there's something there, there's some difference, but it's not particularly informative. So, but what this data is actually informative about is trying to do individual prediction of clinical outcomes. And this is some hot off the press results from Sue. So this is predicting change in the ARCADS measure between the baseline and the one-year follow-up based on resting state fMRI and diffusion MRI measures from the baseline scan. So this was resting state connectivity from Sue's favorite area of DLPFC and subgenual ACC, and fractional anisotropy from the uncinate fasciculus, which is associated with anxiety based on several prior studies. And yes? No, no. Some of them may be medicated, so there are some medications that are allowed just to be able to recruit subjects, and some that are not, but yeah. So what she has shown is that if you use the ARCADS measure from the baseline, you can explain some of the variance in the change between the baseline and the follow-up. But as you add the different modalities, you get better and better predictive power. So this is adding only the diffusion MRI measure, this is adding only the resting state, and this is adding both. So this shows that there is different information included in these measures. And so in some of the studies that have tried to combine functional and structural connectivity, the focus is on how these things are correlated. But I would argue that perhaps the interesting information is how they're different and what complementary information they carry. And as I mentioned earlier, this data set, it should become available somewhere in the middle of the upcoming year. So it includes a lot of, like something like 4 hours of clinical interviews, as well as a 2-hour MRI scan that includes resting state, diffusion, and some tasks also. Okay, so I talked about the good. Now a little bit about the bad and the ugly. So we have improved our tools for acquiring and analyzing diffusion MRI data so much over the last few years. More and more people, or more and more centers are acquiring these new scanners with stronger gradients that are designed specifically to acquire high B-value, high SNR diffusion MRI. And because of that, we can get very accurate estimates of those vectors that I showed you, the diffusion orientations. However, spatial resolution is still limited to about 1.5 millimeter at best. And this means that there's going to be more than one axon population within one voxel. And unfortunately, we are unable to resolve certain architectures, certain configurations of axon bundles within a voxel when we have more than one axon population per voxel. So this is 3 different ways in which 2 axon populations of a blue bundle and an orange bundle can be arranged inside a voxel and give the same diffusion profile. So the diffusion orientation function would tell you that there's some water molecules moving this way, some water molecules moving that way, but this is the case for all 3 of these orientations. And so when a tractography method reaches that voxel, it doesn't know which way it should turn because it doesn't know which of these 3 is the ground truth. So even if you have perfect estimates of these diffusion orientations, you still have that ambiguity in there. And because of that, sometimes it'll turn the right way, sometimes it'll turn the wrong way. So it has to make an arbitrary decision, and inevitably things will go wrong sometimes. So what happens in that case is that the tracks are following one specific pathway, like the corticospinal tract, but then because of taking the wrong turn somewhere in one of those intersections, it jumps onto a different bundle that is actually not really anatomically connected with the first one, but just happens to overlap over part of their trajectory. And, you know, keep following it all the way to the other hemisphere and jump to the corticospinal tract on the other side. Now, for something like this, it's easy, even for someone who's not a trained neuroanatomist, to see it and say, okay, this is probably not right. But for most of the stuff that we see in a whole brain diffusion data set, we have no way of knowing, is this right or is it wrong? And because of that, we still need ground truth from post-mortem validation studies to figure out what goes wrong, where it does, and potentially how to fix it. So in our lab, we have been going about post-mortem validation of diffusion MRI in a couple of different approaches. One is working with Suzanne Haber's lab on macaque chemical tracing, where she does the tracer injections and sends us the post-mortem fixed brain. We scan it in an MRI scanner, and then she does the histological analysis to visualize the tracer, and we can combine side by side in the same brain diffusion tractography and chemical tracing. This gives us access to the fiber population that originates in the specific area where the tracer was injected, but it does not give us access to everything that crosses with that. So there is information there that we can compare to, let's say, doing tractography in the seed region, but we do not know if things go wrong, what caused them to go wrong. A complementary approach that we're following is doing microscopic resolution optical imaging in human samples. This approach gives us orientations of axon bundles, of all the axon bundles that go through an area of the brain. So this is an example where we have a split of fibers between the internal and the external capsules. But at this moment, it is only feasible in small samples, so not in the entire brain. So these two approaches have complementary strengths and weaknesses. And I'm going to go over some of the things that we have learned that have applicability to how we should acquire and analyze our data in vivo. So using the macaque data, where we have chemical tracing and ex vivo diffusion MRI in the same brains, we, in addition to our own studies, we also organized an open challenge where teams from all over the world applied their tractography methods, and accuracy was determined by comparing the tractography to the tracer data. Although there have been some challenges before, this was the first time where we had very high angular resolution and high B-value data. So we could consider both the two-shell scheme that is similar to the one used by the Human Connectome project, as well as a DSI sampling scheme that collects many more, actually 514 different directions at many more B-values, all the way up to a B-value of 40,000, which for postmortem tissue would be the equivalent to having a B-value of 10,000 in vivo. So we had the teams first go through a training phase where they ran tractography results and uploaded them at multiple different thresholds. And then comparing that tractography to the ground truth from the chemical tracing, we built ROC curves of true versus false positives. And the contestants then could see the results compared to other teams, and they could tune the tractography methods to achieve as high performance as possible. And then once that optimization phase was over, we had them apply the optimized methods to a different injection area in a different macaque brain. So the question there was how similar is the optimal method for one injection versus another, one injection area versus another. And these were two different parts of the frontal cortex. So the training phase was in the frontal pole, and the validation case was in the VLPFC. And what we found was that actually for almost all of the methods, the performance dropped substantially in the validation case. There were only a couple of exceptions that were very robust, and we're optimizing for one seeding and injection area led to optimal results for a very different injection area. And so we're in the process of further analyzing this data, working with all the teams to figure out what it was about those methods that did so well. Was it the pre-processing? Was it the tractography? Was it some post-processing that they did? And then come up with recommendations for how the HCP style data should be analyzed. One thing that we found that was very interesting is that actually having the second shell or having very high B-value data, or even comparing the two shells to the full DSI data set of all 500 directions, actually didn't make a huge difference. So it seems based on our results that getting all those super high B-values or even multiple B-values is not that beneficial for tractography. Now, this doesn't mean that you should go back to just collecting a single shell and that multi-shell data is useless. It's actually super useful for a lot of microstructural models like diffusion kurtosis, orientation dispersion type models, et cetera, that require the multiple B-values. But at least based on our results so far, it doesn't seem to make a huge difference for tractography. Similarly, with a very different approach, looking at the optical imaging data compared to diffusion in small human samples. So this is the ground truth from the optical imaging data, and this is the orientations from the diffusion data that was acquired at 1/4 millimeter resolution and then downsampled all the way to 2 millimeter resolution. We find that across different reconstruction models, which is the different columns, and especially across reconstruction methods that use orientation distribution functions, there isn't that much of a difference in the accuracy. So things look fairly reasonably well matched to the ground truth. The ball and stick and tensor model did not do as well, but also across the models, what we saw is that as we went up to a voxel size of 1 millimeter, the accuracy remained pretty constant. But then really where things deteriorated was between 1 millimeter and 2 millimeters. And this suggests that spatial resolution actually does make a big difference. So it perhaps suggests that if you have a certain amount of time to use collecting diffusion data and you're specifically interested in tractography, maybe you should spend more time collecting smaller voxels rather than collecting hundreds and hundreds of directions or super high B-values. This work is in review at the moment as well. So to summarize, what we find from our postmortem validation studies is that there's a lack of dramatic improvement as we go to higher or multiple B-values. And we also know that we will always have this ambiguity and this inability to resolve certain types of axon bundle configurations just based on looking at their diffusion profile. So just based on looking at the orientation distribution function. So now the question is this an inherent problem of diffusion MRI data and we should just give up? Or is it a problem with reducing the full data set into these orientation distribution functions? So these models were developed to model crossings. So for a very long time, the field was focused on solving the crossing fiber problem. And unsurprisingly, these methods are very good at resolving crossings, but they are bad at distinguishing crossings from other types of architectures, like things that split, or anything other than a crossing that involves multiple axon bundles. So I would like to be hopeful and say that this is not an inherent problem of diffusion MRI that we cannot solve, but we just have to go back to the drawing board. And now that we have much higher quality data than we had when these methods were first developed, we have to come up with better algorithms to extract all the information from the data. And I will leave it at that. I would like to acknowledge Robert and Chiara here doing preparing for one of the optical imaging scans on some fellow's dead brain. Hui Wang, who develops the optical rig that we use for those experiments. My other collaborators, Bruce, Jean, and Martin on the postmortem validation, and of course, Suzanne Haber and Julie Lehman from the University of Rochester, who have generously given us access to their monkey data. And just as a broad overview of what I talked about defining bundles based on relative position to other structures in native space, rather than the location in template space. How this principle can be applied both to atlas-based tract reconstruction, where you know the exact bundle that you're looking for, or for data-driven tract clustering when you're doing a more exploratory study. And I mentioned some insights from our postmortem validation studies where we have found that high spatial resolution may be more critical for tractography than having multiple or high B values. And finally, this points to a need for new methods to take better advantage of that multi-B and high B data. And I will end here, and thank you for your attention. Any questions? Yes.
Really amazing.
Oh, thank you.
So the last piece where you spent a bunch of time talking about the ambiguity in crossings and divergence.
Yeah.
How much can that be resolved by combining with resting state?
So, I mean, resting state has its own artifacts, first of all, and which are different from diffusion. And so that's one thing. And another one is that we can have regions that are functionally correlated, but they're not structurally connected with each other.
Absolutely.
So when I am somewhere in the middle of the brain and I see one of these things, it would be hard to tell what parts of the brain are functionally involved in the bundles that go through there. Because especially when I'm doing the exploratory kind of like step-by-step sort of tractography, I know where I came from, but I have no idea where I'm going. So it may be possible to use resting state data to, let's say, get rid of outliers in the diffusion data. But then the question is always, so the paradigm is sort of like the diffusion gives you the roads, a map of the roads, and the function is how much traffic there is on these roads. So by one snapshot of that traffic, can I really reconstruct the full map of the roads? To some extent, yes, but you know, I'll never be 100% sure. It's a very difficult problem. Yes.
I have a question about TRACULA over time points.
Oh yeah, the
TRACULA
longitudinal one, yeah.
studies. So my understanding is that when one of the advantages is you train it on both time points at the same time, so there's a one-to-one correspondence throughout the region, right? How does that work if you are continuing a longitudinal study? For instance, let's say you get the first 2 time points, and you trained it and you run it there.
Yeah.
But the 3rd time point, do you then have to run all 3 at the same time?
I would say yes.
Okay.
Yeah, because if you do it independently, you're losing some of the benefit of that. Yeah.
Okay.
Yeah. Yeah, I mean, that's a problem with things like ABCD where, you know, they'll keep adding time points over 10 years, and then, you know, we have to go back and run longitudinal freezer and longitudinal TRACULA every time. Yes, it is, it's a pain, but actually that's the only way to take advantage of, of the benefit of this approach. Yeah.
I have a somewhat related question, but thinking more about the TRACULA.
Okay.
So, I guess, do you think the issue that we were speaking on last with, you know, millimeter size of boxes, is that an increasing problem when you're dealing with sort of younger,
yeah, so there's a lot of things that get worse here. One is obviously the size with respect to the voxel size. Another one is that there is less diffusion contrast because there's less myelin. So there's even less information to go by to figure out what that preferential direction of diffusion is, just because there's fewer obstacles to the diffusion. So there is diffusion and isotropy even with no myelin at all, and people have shown it in animal studies. But there's a large chunk that's accounted for by the presence of myelin. So the contrast is lower, the resolution is more limited, motion, basically everything is against us.
It does make you wonder, though, maybe if you followed them long enough and then you sort of had a retrospective at an age range where the results were a little better.
Yeah.
very interesting thing.
Yes.
So I have two questions. So I'm wondering if there are, well, first of all, are there any like major problem spots that you've noticed in your higher resolution stuff?
Yeah.
And then my second question is, are there any instances that you can think of where exploratory adventures in diffusion imaging can help inform tracer studies versus the other way around?
I mean, it can help inform what areas to focus on, perhaps. I wouldn't say that I would use diffusion as, you know, the ground truth to determine if tracing is correct. You know, if I said that to Suzanne, she would kill me.
But if you found like a weird looking connection that you wouldn't expect, have you then gone back to Suzanne and been like, hey, can you like put a tracer here and see where it...
Right. So, well, where she puts the injections is very much driven from her interests in psychiatric disorders and OCD in particular. So because of that, most of her injections are in the frontal cortex. So there's basically nothing in visual cortex. But the good thing about having these injections in the frontal cortex is that you have these tracts that travel through the brain in many different planes. So rather than, let's say, the corticospinal tract that mostly goes inferior superior, you have things that turn in pretty much all, like in X and Y and in Z, and that they have to then go through these very tight passages like the internal capsule, the external capsule, etc. So you have a lot of, it's a much more challenging system, I would say, for diffusion than for, the motor cortex or visual cortex or something like that. Yeah, so in terms of the trouble spots, so one problem is that with the tracer, we only see the one bundle, right? So we have to obviously combine information from multiple cases that are sometimes in different animals, etc. So there are some issues with that. But actually, if you look at how the people who do the tracer studies describe the anatomy of these bundles, and then see where the trouble spots are for diffusion tractography, a lot of them are in these places where you have a bundle that's coming down from the cortex and then splitting into two. Like for example, in that injection that was in the frontal pole, there's one bundle that splits to go through the corpus callosum, then it goes through the internal capsule, splits into the external and extreme capsules, etc. Some of it has to then turn and go into the uncinate fasciculus. And we find that a lot of the problem spots are in those splitting areas. And that's especially a problem because the crossing fiber models are just not designed to capture a three-way split. So in the presence of that, let's say here, where you have the fibers coming from down here and splitting into this and this, it wouldn't know that it should split from here to here and here to here, but not, let's say, from here to here. So that's where all of these ODF type models would just fail miserably. And something that we're working on at the moment is a different approach that doesn't go through ODFs at all, and tries to learn different patterns of connectivity within a voxel directly from the diffusion data using deep learning. Yeah, but it's a little bit too early for me to show in this talk. Yeah. Yes.
So, crossing fibers have probably facilities that can be identified in quality control and have to exclude subject. I think that's always frustrated neurons with tractography, is when you have sort of a fan in, fan out kind of free roads that are dollar time. I've never worked with really high resolution data.
Yeah, so something like that is where the higher spatial resolution would be in my view, more important than having many directions or many b-values, especially when you have these fibers that are coming from multiple orientations and then they all get crammed into one narrow passage. So first of all, ODFs are not good at capturing that kind of thing. And second, the voxel size is critical for being able to separate them. So yeah, so if you were interested in looking at something like that, you would probably not go with, you know, trying to get 300 directions with 3B values. I would just recommend, you know, just trying to get the spatial resolution as high as possible. And even if you just do like a B equals 1000 acquisition. To some extent, yeah. I mean, there will always be a limit, right? But because you cannot go too high. There are new acquisition methods, new sequences that are being developed specifically for doing diffusion at sub-millimeter resolution in vivo. And those are still at the research level. So there's nothing that would be available right now as a product sequence, for example. But I think that if we can show the importance of the high spatial resolution with these postmortem studies, I think that will generate more interest and more demand for those kinds of approaches. So those approaches, because of SNR issues, and time, you would not be able to use them to collect, you know, hundreds and hundreds of directions. But ultimately, if you're interested in something like that, yeah, there are some solutions in the works right now.
So much.
Thank you.